Hi, my name is Dalton Williams, and this is BY210 Genetics. So, DNA replication starts out as a double-stranded molecule of DNA. It's antiparallel and complementary, meaning that thymine pairs with adenine, guanine pairs with cytosine, so on and so forth. Antiparallel, one strand is 5' to 3', the other is 3' to 5'. Moving on, DNA, it needs to be broken to replicate. It needs to be broken into two different strands, obviously to replicate one strand and the other. Gyrase comes in here and it relieves some of the stress of the double helix so that the helicase can come in behind and break these individual bonds and split the nucleotides separately. And then once you have all it's come through here and it has broken all of these individual base pairs, then you can have replication begin. Now once these strands are broken, you have one strand from it being antiparallel that is 5' to 3', and the other one that is 3 to 5. Now DNA polymerase, it moves in a 5' to 3' fashion. So you're going to have one strand that is the leading strand and synthesized in the 5 to 3' direction. Then you're going to have a lagging strand, which is synthesized discontinually in a 3 to 5' fashion. So the DNA polymerase, it goes through and it makes the leading strand as the replication fork opens. And then you have the lagging strand, and you have individual DNA polymerase comes in and it makes little tiny 5 to 3' sections, and then it skips over and it starts another it has another catalyst that comes in here and starts and just continues on until a whole strand is synthesized. But then you're left with small little sections like right in here that are just open and there's no base pairs in here. So then you have Okazaki fragments, which are already pre-coded, and they come in here and fill this void like right here. So that is, and this is DNA replication, and that is in a eukaryotic cell. So moving on to a prokaryotic cell, you start out, and instead of the DNA being linear, it is circular. So you start out right here, and it has to start at the origin of replication, which is shown right in here. And then it starts kind of getting pulled back, and you create a replication bubble, which is right here, and then you have a replication fork, and you have the same thing going on where you have one synthesized in a 5 to 3' fashion and one strand discontinually synthesized. And as you go through, you start to form a new strand of DNA between right here in the replication bubble. And then you move on, and eventually these two kind of pull apart, and you're left with two sister chromosomes, and therefore you're replicating prokaryotic DNA. Moving on to transcription. Transcription uses the base pair uracil to pair with adenine instead of thymine, as in DNA replication. And DNA transcription, the role is just to make mRNA, and that is to be a basically to have interaction between the DNA and the outside of the nucleus. It has 3 stages, and that is initiation, elongation, and termination. It has to have a promoter region, an RNA coding region, and a terminator region. In bacteria, the core enzyme and a sigma factor bind together right in here to make a holoenzyme, which will recognize the promoter region and bind to it. It forms an open complex that allows the RNA nucleotides to attach to the DNA template strand and start forming an unconnected RNA strand off the parent strand, and it goes in a 5' to 3' direction. Initiation is over when the sigma factor falls off the parent strand, and elongation continues the process. RNA polymerase moves down the DNA and is wound back up, and termination ends when the whole strand has been converted to mRNA. Now, the difference between the prokaryote and the eukaryote is that the eukaryote has the 5' methyl G cap and the 3' poly A tail. Now, right in here, you have parts that do not need to be in here for transcription to occur. So you use spliceosomes to kind of pull out all of the introns that are in here, and then you are left with the exons, and transcription can continue. The second step of protein synthesis is translation. There are 3 steps to translation: initiation, elongation, and termination. A small subunit of rRNA bind to mRNA in the Shine-Dalgarno sequence, which is right here. After attachment, the ribosome scans the RNA right in through here for the initiation codon. Once the codon is found, an anticodon attaches. This is the small subunit, attaches, and then it calls for the large subunit to attach. That is the end of initiation. Then you begin elongation, and in elongation we're pretty much dealing with the large subunit, and you have the E spot, the P spot, and the A spot. An anticodon comes in and attaches to the P spot to begin with, and then you have another polypeptide, you have another amino acid that comes in and attaches at the A spot, and then you form a polypeptide bond between the amino acid in the P spot and the amino acid in the A spot. The amino acid that was in the P spot no longer has the polypeptide bond, so it moves to the E spot. The amino acid with the polypeptide bond now moves to the P spot, and another tRNA comes in to the A spot as the tRNA that was in the E spot leaves. And then in termination, after everything, coming back to right here, after everything is gone and a nonsense codon is reached, a release factor is attached, and the codon and the entire complex, so right in here, releases, and then you are finished with translation.